My name is Ede Lars. I'm from Delft University of Technology in the Netherlands. And today I'm going to give a presentation on how we used Spark as a framework to enable well-known, state-of-the-art personalized medicine pipelines, computational pipelines, to run at scale on computational infrastructures, enabling less than $100 and less than 1 hour computational capacity for our pipeline. Just to start the discussion, I would like to show the development of computational and DNA sequencing information in the past 15 years. On the top line, you see Moore's Law, which represents the reduction in cost of computation in the past 15 years for computers. And on the bottom, you'll see a similar graph that shows the reduction in the cost of acquiring DNA data. And if you see from this figure, of course, Moore's Law is an exponential decrease in price. And you see below that DNA sequencing data, so acquiring DNA data, has been reducing at a much faster rate than the exponential rate of Moore's Law, enabling a number of different applications in personalized medicine, such as using targeted therapy for genetic diseases such as cancer and otherwise. The problem with this figure, the challenge that is introduced with this kind of reduction, fast reduction in cost in acquiring DNA data, is that at some point at 2007, 2008, 2009, the cost of acquiring DNA data has went below the cost of actually computing and getting knowledge and insights from this information, which has shifted the challenge from the actual acquisition of the data to the computational part of the data, which we would like to introduce a solution for. One of the main reasons for having this computational challenge is based on the huge data sets that are being used in the area of personalized medicine. We're talking about gigabytes to terabytes of data for a single experiment or measurement sequencing experiment for a human being, and also the huge computational characteristics or requirements for the analytics pipelines that are being used in practice. Here I show an example of one state-of-the-art, what we call variant calling computational pipeline. So variant calling or variant discovery pipeline is basically a personalized medicine pipeline, DNA analysis pipeline, that actually identifies mutations that are taking place in a DNA sample of a cancer patient, for example. So this pipeline has 3 main stages that I will keep on referring to throughout my presentation. The first stage is what we call DNA mapping stage. In that stage, a number of different reads or number of different representations of the DNA, snippets of the DNA of a patient are being mapped or located on a reference DNA to identify how the patient's DNA looks like. The second stage is what we call the sorting stage. In that stage, the location of these different snippets, DNA snippets, is being sorted. And the third stage is the GATK pipeline, which is a well-known Broad Institute pipeline that enables identifying the mutations in the patient's genome as compared to a normal genome. The input is a FASTQ file or a DNA information file acquired from the sequencing machine, and the output is a VCF file or variant calling file that represents mutations in the DNA of a patient. If we look at the profile of executing such a state-of-the-art pipeline on standard computing systems, you will see that in this figure, you will see that only the first stage, the mapping stage, which uses a program called, state-of-the-art program called at the moment BWA, only the first part of the pipeline is actually achieving close to full utilization of the computational infrastructure available for an institution. The other stages of the pipeline, you will see that many of the curves stay low or have some spikes that go higher, enabling higher utilization of the infrastructure, but then falter down and uses very low capacity of the available infrastructure, and sometimes even sequentially using a single core and single thread of the immense computation capacity that an organization have. The challenge that we see is enabling an easy, accessible way to be able to utilize more of the capacity of the computers that you have to enable faster and cheaper computation of the pipeline. Now, the idea that is commonly used to enable this kind of solution is basically by parallelization. So what happens, we know that different chromosomes in a human being are relatively independent from each other. So what researchers do basically is that they run multiple pipelines, multiple variant calling pipelines in parallel, and they run them on different genomes, different chromosomes of the genome independently or in parallel. Now, running this kind of pipeline in this fashion will enable up to 22, 23 kind of parallelization, because you have 22, 23 chromosomes in your genome. But we also discovered that it is possible to perform more parallelization by breaking some of the chromosomes in parts and running even higher parallelization by breaking the bigger chromosomes, for example, chromosome 1 or 2 or 3, in more parts to enable higher parallelization. And it proved to be quite effective and and ensures very high accuracy of the analysis, as I will show later in the presentation. Now, there are a number of different techniques that are being used today to enable utilization of the infrastructure for this genomics pipeline. Now, there is the parallelization infrastructure that is provided by GATK pipeline itself, which is based on the grid engine, the Sun grid engine. The pipeline has some kind of disadvantages. First of all, it's hard to set up and difficult to use by professionals in the field, and it uses rather slow scheduling, has disk-intensive access, which limits its advantage in using your computational capacity. There are also a couple of solutions that depend on Hadoop, which, as we know, has intensive disk access as well, and it has limited memory utilization, which limits the capabilities of your pipeline of utilizing advanced techniques to increase the capacity. There is Atom as well that uses Spark, which has in-memory computation, but it lacks actual validation from the genomics environment. It uses its own API that requires geneticists to develop specifically on Atom to enable some kind of solution. Now, our solution is actually uses state-of-the-art techniques, so existing computational pipelines developed by renowned universities and research centers and clinics, and we simply enable that solution using Spark as a framework. So easily plugging in your pipeline into our Spark framework will enable very easy and transparent scalability on the infrastructure that you're using. To give you an idea of different stages that we use in our Spark framework, basically we start with a pre-processed form of the input, creating a number of different parallel chunks of the FASTQ or the DNA snippet files. And then we go through 3 different stages that represent the 3 stages of the pipeline that we showed earlier. So we start with a mapping stage, and we do a little bit of static load balancing, as I will discuss later. And then we go to the sorting stage, and then we do some dynamic load balancing, as I will discuss later. Here we use effectively the in-memory computation capabilities of Spark. And then the third part, we run many parallel GATK pipelines on many different regions that we create within the genome of a patient. And we end up with a VCF file that is a joint of all different VCF files that have been generated by different chunks of the pipeline. I'll go a little bit in more detail in each one of these stages, just to give you an idea of what we have done without going into all the details of the code and everything. So pre-processing is rather an easy and straightforward approach process. We just merge the files, the input files together, and then we divide them segment them into chunks depending on the amount of parallelization that we are having, that we have in our infrastructure. So you have 10 nodes that have 10 cores each, with each of them having multiple threads. You will just multiply 10 by 10 by 2 if you have 2 threads, and then run those in parallel. And that is easily enabled by the mapping program itself, because it's a easily scalable program in the that can be run on multiple threads and multiple nodes. That happens in the first stage, which is the mapping stage. We use the BWA program, which is a well-known program that is used almost by every personalized medicine institution to do the mapping, and also by other kind of applications for DNA analysis. And the BWA program actually is able to scale easily, as I said, into a scalable infrastructure. And in addition to this, we enable, in addition to using BWA to run multiple parts of the program in parallel, we also, as the output comes out of the BWA, which is a mapping of the different DNA snippets to a reference genome, enabling us to know the location of the different snippets on a reference genome, we also keep those mapped files for different chromosomes separate. So basically, each BWA task, the output of that is separated into smaller chunks, each one representing the location of the mapped snippets of DNA to different chromosomes, and sometimes to parts of these chromosomes if we see that there is benefit of using higher parallelization. The second stage is basically we bring all these files together, taking the output of each chromosomal region or each chromosome at a time. That will enable us to optimize memory utilization. So usually these files are really huge. We're talking about gigabytes, hundreds of gigabytes to terabytes of data. So usually a computational node does not, the computational nodes do not have this amount of memory. So we can, depending on the capacity of the memory that you have in your node, we are able to take parts of this data, put it in memory, and do what we call dynamic load balancing. Basically also looking dynamically at the amount of reads that you have in specific regions of the DNA that you have mapped. And then if we see that some chromosomal regions have higher number of reads as compared to other chromosomal regions, we'll go on and split those into further sections, enabling what we call dynamic load balancing, distributing the load on the network more in a more balanced way, enabling better utilization of the infrastructure. And then we do the sorting after that, and then divide the output into various chromosomal region files, and then we basically bring all these together and run them on multiple different GATK pipelines in parallel. And then we choose the number of these pipelines depending, again, on the amount of parallelism that you have in your infrastructure. Basically, the number of, we choose a different number of Spark containers, and this number of Spark containers depends on how much parallelism can you enable in your infrastructure. The more you have, of course, the better parallelization you can achieve, and the faster your solution can be calculated. And the output is, again, collected together into a VCF file. That is the framework of our solution, and then we wanted to set out and implement it on a large cluster to find out how scalable it is and how much do we benefit from the utilization of an infrastructure. To do this, we chose an IBM Power 8 system that enables extreme parallelization for using the benefit that it has by having a large number of multi-threaded capabilities for each core, a large number of cores that are running on each processor, and multiple two processors that are enabled in each node, having really a very aggressively multi-processing kind of environment in every single node. We have 20, in our largest system that we use, we have 20 Power 8 nodes. Each of them has 2 different processors. Each processor enables 10 processing cores, and each core is able to achieve 8 simultaneously multi-threaded threads computed in parallel. which means that every node itself is able to run up to 160 threads, hardware threads, in parallel. Having 20 nodes, we're talking about 1,600 to 3,200 threads for the cluster that we have used in our system. I also mentioned the kind of input data that we have used. We have used here a 400 gigabyte genome, whole human genome sequencing data set to be able to see what the impact of this large data set on the experiments. Some of the results that I would like to show here, this is basically a profile of a run that we have executed on the Power 8 cluster. I show the profile of a single node here, and the red line basically shows the utilization of the node and the execution profile of the GATK pipeline on this node using the Spark framework that we have produced. Now, if you look at the red curve, you will see that actually most of the time the red curve is hitting almost 100%. And there are some times where it goes down, spikes that go down, and then there is in the middle some part that is not very well optimized. But if you compare this kind of figure to the figure that we started with in the beginning, you will see that it is almost the opposite. Most of the time, we're fully utilizing our infrastructure, and sometimes we are waiting for disk or waiting for for communication to take place. The scalability can be shown in this figure. On the x-axis, you will see an increasing number of nodes that are being used for our Spark infrastructure. So we see 8, 10, 14, 15, and 19. And of course, we have one node that is the name node that controls all other nodes in our Spark cluster. And you will see that there are 3 different curves in there and one total curve. The 3 different curves represent the 3 different stages that we have in our Spark implementation. In this figure, you will see that stage 1, the first curve on your right-hand side, actually scales down appropriately very well as we increase the size of a cluster, almost linearly, starting with about 40 minutes, a little bit more than 40 minutes, and ending with a little bit more than 20 minutes as we move from 10 to 19 nodes in the cluster. The third stage is as well rather scalable. So we're moving somewhere from 100 minutes to all the way down to 50 minutes as we move from 8 to 19 nodes in the cluster. The only part that is a little bit less parallelizable, scalable, is the middle part that you see it goes down a little bit from 8 to 10 nodes and then stays down around 20 minutes as we scale up the size of the cluster. And this, of course, reflects the slide that we just showed earlier. The first and the last part of the pipeline have very good utilization of your infrastructure, and only the middle part, stage 2, that has a little bit less optimal utilization. Now, if you look at the profile of various nodes in the cluster, we'll see that this kind of profile is also reflected in almost all the nodes that we have in our infrastructure. So most of the nodes have the same kind of profile. They have a lot of utilization in the beginning, a lot of utilization in the end, and then a little bit less utilization in the middle part. We also show here the I/O characteristics for these different nodes, so how they access disk. And we see that most of the time, actually, the Spark infrastructure is not fully utilizing the available capacity of disk access at all. So it is not I/O bound either when you run these profiles. Now back to our promise. We started the talk by saying that we will be able to enable whole genome sequencing that usually takes days or weeks to be run within less than 1 hour and with less than $100 as cost. Now, this figure shows how we can scale our solution up to less than 1 hour. Our 19-node, 20-node cluster was able to achieve 90 minutes scalability runtime for the whole human genome processing pipeline. Now, what we did with this figure is that we took the stage 1 and stage 3, which are very scalable. we extended their scalability characteristics while increasing the size, the number of the nodes in a cluster, and we kept the middle stage, which did not scale very well, at the same number of minutes that have been shown for the 20-node cluster. And we see that if we increase the size of the cluster up to 35 nodes, we are able to actually hit that 1 hour processing time computation for our full whole genome sequencing experiments. Talking a little bit about the accuracy of our analysis, we see that the analysis using Spark, while dividing different chromosomes into smaller regions, still achieves quite high accuracy level as compared to a typical normal serial pipeline. So we see here concordance level, which means the number of mutations that have been identified by both pipelines are almost concordant. Up to 99.1% of all mutations have been identified exactly the same. The GATK specific pipeline, the serial one, has only 0.2 to 3% extra mutations that have not been identified by our Spark-based framework. And just need to mention that regular GATK pipeline itself is not very deterministic. So if you run the same GATK pipeline twice or 3 times, you will get a little bit different results as well, which are not represented in this figure. And the final slide I would like to present is with regard to the cost estimate. We based our cost estimate on cloud service provider costs of a Power 8 node in soft layer. And we see if we take a range of different Power 8 nodes in soft layer in a cloud provider, it ranges somewhere between $30 to $70 per node per day. Now, if we take our solution and scale it up to 35 nodes, we will see that we are able to achieve the 1 hour calculation time, less than 1 hour calculation time for 35 nodes. And these are multiplied by the $30 to $70 range, is well below the $100 computation that we promised in the beginning of the talk. In conclusion, I would like to present you with a Spark, a framework that enables easy and scalable acceleration and speed up of a GATK pipeline on a scalable infrastructure without sacrificing accuracy. We were able to calculate a whole genome sequencing data set of 400 gigabytes under 1 hour with 35 nodes, and we were able to achieve it with a price point that is below $100. Future work would be on optimizing some of the stages that were not very optimal in the pipeline that we saw earlier, and also adding some more higher-end acceleration techniques like GPU or FPGA acceleration for various nodes in the pipeline using the CAPI interconnect that is enabled by the Power 8 nodes. I would like to thank a couple of people that enabled this work. Hamid Mushtaq, who is my student, my colleague in Delft University of Technology in the Netherlands, and a bunch of people from IBM that helped us make this work possible. Peter Hofstee, Frank Liu, who led the effort from the Austin Research Lab, Raj Krishnamurthy and Indra Dijpudar, IP, who actually led the work in applying the Spark implementation on the infrastructure in IBM, and a number of other people who also help with optimizations. Thank you very much for your attention, my contact information, and let me know if you have any questions.
Excellent. All right, please say your name and where you're from.
Hi, I'm Xiangwei from Databricks. So I saw at the end of your pipeline, you put all the files together into a single VCF file. So how big is that file, and does it cause scalability problems?
That file is actually, the VCF file is not a bottleneck in the pipeline. So it's rather small. It basically identifies mutations in the genome, which as compared to the input file, hundreds of gigabytes, we're talking about a couple of megabytes. So that's not really big and doesn't cause any limitations in the pipeline.
Hi, thank you for your talk. I'm Di Wu from UCLA. I was wondering in your pipeline, the stage 3, so all the GTK pipeline are distributed among different chunks of the input data. So do you need any synchronizations between those chunks? Because some of the steps in GTK, like the base recalibration, that requires you look at the entire genome to calculate the more accurate results. So I was just wondering how you do the synchronization. Thanks.
So that's exactly the part of the pipeline that actually requires you using the data from different regions and also different chromosomes to be able to calculate accurately the kind of variations or mutations that you have in the pipeline. Exactly. So by actually splitting the data set into a number of different chromosomal regions, we are actually not enabling this kind of calculation to take place across chromosomal regions. So you're right, we are, by this dividing the data set into chromosomal regions, we are preventing actually this kind of synchronization. So we are not taking care of the synchronization, and that also results in the eventual difference in the accuracy between the Spark pipeline and also the serial pipeline. We showed by a number of different experiments that this accuracy is actually quite high, and it does not require us to make sure that this synchronization is respected to ensure high accuracy. Of course, if you would like to ensure high accuracy, that is possible, but then you will have to sacrifice a little bit of the utilization and performance in the pipeline as well.
Have time for another question? All right, let's give Zaid a big hand.
Thank you.